All right, ladies and gentlemen, welcome to our first screencast of week 2 for cell division. So far, we know that the cell spends the majority of its life in a phase of the cell cycle known as interphase. Interphase, we know, is collectively made of individual stages, those stages being G1, S, and G2. And after interphase, think back to your pie chart on your study guides, after interphase comes either mitosis or meiosis, depending on the type of cell. And after mitosis and meiosis comes cytokinesis. But mitosis, remember, has 4 distinct phases all its own. They are pro, meta, ana, and telophase. Now, we're going to start looking at anaphase, the third of 4 steps of mitosis. If you recall back from metaphase, all of these chromosomes you picture here were lined up across the middle, right? And they're still kind of in the middle now, but a lot looks different here in anaphase, right? And let's recall that these centrioles at the poles connected to these spindle fibers connected directly to the chromosomes at the centromere. Okay, let's not forget that. So in anaphase, the centromere is basically divided in half. That way, one sister chromatid goes to one end of the cell and the other sister chromatid goes to the other, right? So if we take this chromosome here in the middle, for example, it's attached to a spindle on this end, it was attached to a spindle on this end. As each of these spindles shortened, it pulled this duplicated chromosome apart at the centromere. So now, at this point, this is a single chromosome and this is a single chromosome, comprised of only one chromatid. And you can see they almost look like they are moving toward opposite ends of the poles, because they are. So we have chromatids being pulled to opposite sides of the cell. And these microtubules, right, these yellow arms, these just protein structures, are shortening. And this is what actually pulls the chromatids to the opposite poles of the cell. Again, this is the whole point of the centrioles, this organelle we talked about back in Unit 3, is to produce this spindle complex attached to the chromosomes, organize them, and separate them during cell division appropriately. These are the shortening microtubules. And by the end of anaphase, the 2 ends of the cell should have equivalent and complete sets of chromosomes, right? So if this is a human animal cell, right, there'd be 46 of these duplicated chromosomes. Half of each chromosome would go to each end of the cell. So each end of the cell would have 46 chromosomes in it. Now, telophase is probably where our cell looks the most different. Take a look at it now, I'll give you a second, and kind of analyze what's happening here. And notice the overall shape of the cell. So in telophase, we see a lot happening. The nuclear membrane is beginning to form, right? That's what this is. If we recall back to prophase, this is kind of how we showed the nuclear membrane degrading or breaking down. But in this slide, we see that there's one nucleus forming here and one nucleus forming here around the chromosomes that have been pulled to each end of the cell. So we see 2 nuclei forming within the same cell, right? This is still one big cell. The nucleolus is going to start to be rebuilt. We can see the nucleolus being rebuilt here, and we can see the nucleolus being rebuilt here, right? Because these new cells are going to have to make ribosomes on their own, so they're going to need a nucleolus. Ultimately, a very important aspect of this point in cell division is what's called a cleavage furrow. Okay, that's what this is, right? That's what's starting to happen here. It's kind of the telltale sign of telophase. Like if you see this pinching in, right, and the cell almost looks like the number 8, if you see this pinching in, it's indicative of telophase, all right? This pinching in tells you it's telophase. And actually, this pinching in is cytokinesis happening a little early, right? Cytokinesis doesn't wait until the very end of telophase. As soon as these microtubules, this spindle complex, pull the chromosomes to opposite ends of the cell's membrane will start to pinch in, and this is cytokinesis starting as telophase ends. So we have our nuclear membrane returning, we have our cleavage furrow, our pinching in of the cell membrane, which is a precursor to cytokinesis. And the end result is going to be two cells that are exact copies of each other, ideally. Remember, errors do happen, mutations in DNA can cause this to not be the case, but ideally the goal was to make one cell here be identical to this cell here. Okay, so we'll take some time here and we'll look at numbers 1, 2, 3, 4, and 5. Okay, and we're gonna label these. Your options are either going to be interphase, prophase, metaphase, anaphase, or telophase. So let's look at number 1. Number 1 is right here. All of these are number 1s. Okay. What do you think number 1 is? And these are slides from under a real microscope. These are similar to what we're going to see this week. Number 1 is actually prophase. Okay. Number 1 is prophase. You still see a defined circular or oval nucleus, right? And a telltale sign of prophase is this round-ish nucleus still, and the center is very dark, right? Because the DNA, which is the purple part in these images, the DNA is condensing. It's becoming more visible. So when it's stained, it's more visible. Okay, so this is prophase. Number 1 is prophase. Number 2, let's see what number 2 is. These are your two options for 2. Number 2 is actually metaphase. Number 2 is actually metaphase. So if this, and it's outlined pretty lightly, but this is a cell, right? These are plant cells, by the way. So if this is a cell, the chromosomes, I know they look a little spiky here, but look, there's a general trend across the middle, right? These chromosomes are lined up across the metaphase plate. And same here, this is just a worse state. This is a slide that has been stained more poorly. So it's harder to differentiate. All right, let's look at number 3. What do we think number 3 is? Number 3 is anaphase. All right, if you recall back, go back and look on your study guide right now at the actual illustration of anaphase. These chromosomes look like they're getting pulled to opposite directions of the cell, which they are. Okay, and they look like it. So that's anaphase. Number 4. Now, because this is a plant cells do divide a little differently, right? Their cytokinesis step in particular is different. So we see the rectangular shape of the plant cell thanks to its plant cell wall. But we don't see a cleavage furrow here, right? So we can't definitively say this is telophase, right? Well, this plant cell, you can see there's almost a divide down the middle, right? Or a growing divide. There's this dark line that hasn't made it out either way yet. We call that the cell plate. It's the plant cell equivalent to a cleavage furrow. Another telltale sign is that there are 2 nuclei in this one cell, right? That doesn't happen until telophase, when the nucleus returns. And lastly, number 5 is our only one remaining, and that's interphase, right? See, one cell, and then we have this very lightly stained nucleus. Now, excuse me. We have this very lightly stained nucleus, and the reason the nucleus looks lighter is because the DNA is not as condensed as it will be over here in prophase, right? Because interphase leads to mitosis, prophase being the first stage of mitosis. So this is interphase. It looks lighter, but it looks very similar to prophase. All right, folks, stay tuned.